Hey guys, welcome to the new video. Today I'm gonna cover microbiology. And this is chapter 3, and chapter 3 covers cell structure and function. So yeah, let's just go over it. Now, this whole PowerPoint has over 100 slides, as you can see right here. And so this video may take long. So, processes of life. Now, the question back then was, what does living mean, and how do we define life? And at first, this may seem like a simple question, because after all, you can usually tell when something's alive. However, defining life itself can prove troublesome. So, biologists generally avoid setting a definition, and instead they prefer to describe characteristics common to all living things. And biologists agree that all living things share at least four processes of life, which is growth, reproduction, responsiveness, and metabolism. Okay, so characteristics of life and their distribution in microbes. Now, before I continue, I want to make a comment in regards to viruses. So viruses, they blur the line of living and non-living. However, most scientists consider viruses to not be living because they do not meet the criteria of life. So, for example, growth. Growth basically means increase in size, and it occurs in both prokaryotes and eukaryotes. But if you look at viruses, growth does not occur. Same thing with reproduction. Reproduction means increase in number. Happens to both prokaryotes and eukaryotes, but viruses, host replicates the virus. Um, and you can say the same with the rest that I'm about to show you, which is responsiveness. Responsiveness means the ability to react to environmental stimuli, but for viruses, reaction to host cells seen in some viruses. Metabolism, controlled chemical reactions of organisms. Viruses use host cells metabolism. And cellular structure, in which membrane-bound structure capable of all of the above functions. And viruses lack cytoplasmic membrane or cellular structure. So overall, they are not living by, in definition, to what a life is. Okay. So examples of type of cell. So image A shows a Escherichia coli bacterial cell, whereas image B shows a paramecium, a single-celled eukaryote. And the book says note the differences in magnification as shown by the scale bars. So what this is saying is that eukaryotic cells tend to be bigger than prokaryotic cells. So that's what you have to take away from this slide. Okay. Prokaryotic and eukaryotic cells, an overview. So prokaryotes. The word prokaryote comes from the Greek word meaning before nucleus. And a distinct feature that prokaryotes have is that they lack nucleus. And since they lack nucleus, they can read their DNA genetic code and simultaneously make proteins. Prokaryotes also lack various internal structures bound with phospholipid membranes. They are typically 1 nanometer in diameter or smaller. Prokaryotes include bacteria and archaea. And so here's a typical prokaryotic cell. The artist extended an electron micrograph to show the three dimensions. Note that not all prokaryotic cells contain all of these features. Then we move on to eukaryotes. The term eukaryote comes from the Greek word meaning true nucleus. So by that definition, you can assume that eukaryotes have nucleus. Eukaryotes also have internal membrane-bound organelles. The organelles are cellular structures that act as tiny organs to carry out one or more cell functions. Eukaryotes are larger than prokaryotes. They are typically 10 through 100 nanometers in diameter. Eukaryotes also have more complex structure, and that is referring to the structural plus functional differences between a prokaryote. And eukaryotes are composed of algae, protozoa, fungi, animals, and plants. So here's a typical eukaryotic cell, and note that not all eukaryotes have all these features. Approximate size of various types of cells. So bird egg cells are the largest cells. Then you look at the protozoa, which is the bacterium, Staphylococcus, and the eukaryote, the Giardia. So virus is only there to be shown for comparison. And this PowerPoint is again trying to prove that eukaryotes are typically bigger than prokaryotes. So tell me why. In 1985, an Israeli scientist discovered the single-celled microbe called the Epilopium fisheloni. This organism is visible with the naked eye. So why did the scientists initially think that Epilopium was eukaryotic? And the answer to that was eukaryotes are larger than prokaryotes, so they assumed that it was a eukaryotic cell. The second question says, what discovery revealed that the microbe is really a giant bacterium? The scientists found that no nucleus was found and their external structures were of a bacterial cell, a prokaryote. External structures of bacterial cells. So many cells have special external features that enable them to respond to other cells and their environment. So in bacteria, for example, these features include the glycolysis, flagella, fimbriae, and pili. And starting off with glycolysis. Glycolysis is the plural of glycocalyx, which means sweet cup. It is a gelatinous, sticky substance surrounding the outside of the cell, and it is composed of polysaccharides, polypeptides, or both. And both of the polysaccharides and polypeptides are produced inside the cell and extruded onto the cell's surface. And there's two types of glycocalysis. We first have capsule. Capsules are composed of organized repeating units of organic chemicals. It is firmly attached to the cell surface, and capsules may prevent bacteria from being recognized by the host. We then have slime layer. Slime layer are loosely attached to the cell surface. It is water soluble. Water soluble basically means it can dissolve in water. And the sticky layer allows the prokaryotes to attach to surfaces. So here's an image of a glycocalysis. There's a capsule to the left and a slime layer to the right. So again, glycocalysis are protective layers found in many bacteria that serve multiple functions, including protection against desiccation, aka drying out, and enhancing the bacteria's ability to cause disease. So in summary, glycocalyx helps bacteria survive in harsh environment, evade the immune system, and form biofilms, all of which contribute to their ability to cause diseases. So, for example, encapsulated bacteria can evade immune defenses, leading to diseases like pneumonia, while biofilms such as dental plaque can lead to chronic infections like cavities. And then we move on to motility. So here's a quick animation from the book showing an overview of motility.
Motility is the ability of an organism to move by itself. The motility of a cell may enable it to move toward a desired environment or to flee from a harmful environment. Bacterial cells use structures called flagella, or in the case of spirochetes, flagella-like structures called axial filaments to enable this movement.
So, flagella. Flagella are responsible for movement. They have long structures that extend beyond their cell surface, and they are not all present on all bacteria, but for those that do, their flagella are very similar in composition, structure, and development. So, flagella. Bacterial flagella are composed of three parts. It is the filament, a hook, and a basal body. The basal body anchors the filament and the hook to the cell wall. And then we look at the proximal structure of the bacterial flagella. So for gram-positive bacterial cells, it has a single pair of rings in the basal body that functions to attach the flagellum to the cytoplasmic membrane. Then we look at gram-negative. Gram-negative has two pairs of rings. One pair anchors the flagellum to the cytoplasmic membrane, and another to the cell wall. Then here's another video explaining the flagella structure.
The long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical globular protein molecules called flagellin. Molecules of flagellin form helical chains around a hollow core. Certain pathogenic bacteria can be identified and classified by differences in their flagellar proteins. In contrast with eukaryotic flagella, no membrane covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a curved structure called a hook. The hook is composed of a different protein and connects the filament to the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of either 2 or 4 rings, which anchor the flagellum to the cell wall and allow the flagellum to rotate 360 degrees. You're viewing a flagellum from a gram-negative cell. A gram-positive cell would only have 2 rings in its basal body, both of which are attached to the plasma membrane.
So here is a micrograph of the basic arrangement of bacterial flagella. Now, peritracheous is a term used to describe a cell having a flagella covering the cell surface. So as you can see, there's flagella everywhere around the cell surface. Polar is a term used to describe that the flagella are only at the end of the cell. So as you can see, there's flagella at the ends. Then here's another video explaining the flagella arrangement.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lophotrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Paratrichous bacteria have flagella covering the surface of the cell. Runs occur when all of the flagella rotate counterclockwise and become bundled, while tumbles occur when the flagella rotate clockwise and separate.
Yeah. So axial filament. So image A shows an scanning electron micrograph of two spirochete cells, which is the Borrelia burgdorferi, which causes Lyme disease. Now, spirochete cells have flagella at both ends that spiral tightly around the cell instead of protruding into the surrounding medium. So in summary, this diagram, both of the diagrams actually demonstrates how the unique flagella-based locomotion in these spirochetes enable these bacteria to move through the environment by rotating the axial filament and twisting their entire cell, which facilitates the corkscrew-like movement. This type of movement is particularly useful for penetrating dense or thick media such as human tissue, which is why spirochetes can cause diseases like Lyme disease and syphilis. So spirochetes, this one is another small animation.
Spirochetes are a group of bacteria that are spiral-shaped. They move in a corkscrew-like fashion, using flagella that do not protrude from the cell wall like typical flagella. Lyme disease is caused by a spirochete called Borrelia burgdorferi. Another spirochete, Treponema pallidum, causes syphilis. Spirochetes have an axial filament that lies between the outer membrane and the cell membrane. The axial filament winds around the outer edge of the cell. The axial filament is made up of a bundle of modified flagella called endoflagella. Rotation of the endoflagella causes the axial filament to rotate around the bacterium, which causes the spirochete to corkscrew through its medium. It is thought that this manner of movement allows the spirochetes to move more easily through viscous human tissues and fluids, such as mucus. Then they can easily attach to the cells below.
Okay. So flagella function. The flagella rotation propels the bacterium through the environment. The bacterial flagella rotates in a full 360-degree circle. So think of like a propeller of a boat or a plane. It is powered by ions through the cytoplasmic membrane near the base of the flagella. This provides energy for the flagella to rotate, and it rotates really fast at around 100,000 revolutions per minute. So the rotation allows the bacteria to move very quickly, up to 60 times their body length every second, which is equivalent to a person going around 670 miles per hour. The rotation is reversible too. It can be counterclockwise or clockwise. The bacteria move in response to stimuli or taxis in which it runs and tumbles. So when a flagella runs, the bacteria is moving forward in a straight line, and the flagella is rotating counterclockwise. After some time, the run is interrupted by tumble, which is a sudden random change in direction. And this happens when the flagella rotates clockwise. Okay. So here's the motion of a paratrichous bacterium. Again, paratrichous means that the flagella is covering the cell's surface. So this slide shows the movement of the bacterium. So run, again, occurs when the flagella rotates counterclockwise, and it becomes bundled when it's running. Tumble occurs when the flagella rotate clockwise, and it becomes unbundled, and the cell spins randomly. Okay. Fimbriae and pili. Fimbriae are rod-like protein extensions that stick out from the surface of a bacterial cell. There can be hundreds of fimbriae on a single bacterium. Fimbriae are used by bacteria to adhere to one another and to substances in the environment, which is why fimbriae are sticky and they have bristle-like projections. Fimbriae are also shorter than flagella, and they serve an important function in the biofilms. Biofilms are communities of microorganisms. So going back to the functions of fimbriae, fimbriae help bacteria stick because fimbriae allows bacteria to attach to surfaces like tissues, medical devices, or natural surfaces. Fimbriae also hold bacteria together. After attaching, fimbriae help bacteria stick together, creating a community of bacteria that forms a biofilm. Fimbriae also keep the biofilm stable. Fimbriae help keep the bacteria attached to the surface and to each other, allowing the biofilm to grow and stay strong. And finally, fimbriae protects the bacteria. Biofilm help bacteria survive by making them more resistant to things like antibiotics or the body's immune system. So in short, fimbriae helps bacteria stick, group together, and stay in protective biofilms. So as you can see, this image shows a bacteria, also known as the Proteus vulgaris, which has flagella and fimbriae. And notice the difference between those two. And here's an image of a biofilm. So a biofilm is a community of bacteria surrounded by a protective slime that helps them stick to surfaces and survive. So this picture here shows the inside surface of a medical tubing. Okay. We then move on to pili. Pili is a special type of fimbriae. It is also known as the conjugation pili. Now, the reason why it's also known as the conjugation pili is because cells transfer DNA from one cell to another via the process called conjugation. This essentially helps bacteria share genetic information, such as genes, for antibiotic resistance. Pili are also longer than fimbriae, but shorter than flagella. So pili, it is longer than fimbriae, but shorter than flagella. Bacteria typically have only one or two per cell. So this image here shows two E. coli cells that are connected by a pili. And again, cells use pili to transfer DNA from one cell to the other via a process called conjugation. Okay. So tell me why. Why is pilus a type of Fimbria, but a flagellum is not a type of Fimbria? And the answer to that is Fimbria and pili are used for attachment, whereas flagella is used for motility or movement. Bacterial cell walls. Bacterial cell walls provide structure/shape and protect cells from osmotic forces. So osmotic forces means the pressure difference due to the water flow. And what this bullet point is trying to say is that cell wall prevents the cell from bursting open when water enters, hence why it protects the cell from osmotic forces. Bacterial cell walls also assist some cells in attaching to other cells or in resisting antimicrobial drugs. It can target cell wall of bacteria with antibiotic. So note that animal cells do not have cell walls. We have animal cells. And this has a difference that plays a key role in the treatment of many bacterial diseases with certain types of antibiotics. So, for example, penicillin. Penicillin attacks the cell wall of bacteria, but it is harmless to humans because we lack cell walls. So, what these two bullet points are trying to say is that these antibiotics are basically designed to disrupt or interfere with the synthesis, structure, or the integrity of the bacterial cell wall. And that's because the bacterial cell wall is crucial for the survival of bacteria because it provides structural support, it protects itself from bursting due to the osmotic pressure, and it maintains the overall shape of the bacterium. So as we know, bacterial cell walls gives the bacteria cells characteristic shapes. There's cocci and bacilli. So bacteria that are round in shape is called cocci, and there's three types. Streptococci are chains of spherical cells. Staphyloci are clusters of spherical cells. And then Sarcinae are cube/packets of 8 spherical cells. Now, bacteria that are rod-shaped are called bacilli. Bacilli appear singly, which means they appear individually or alone, or in chains. So the image in the left, or image A, it shows a cocci, or cocci. And when they are in chains, that is called streptococci, or cocci. Now, when in clusters, it's called staphyloci. And then when in cuboidal packets, it's just Sarcinae. Now, the image in the right, or image B, this shows a bacilli. Now, regardless if it's individually or in chains, it's still called a bacilli. Okay. So, the main component of bacterial cell wall is called the peptidoglycan. Peptidoglycan are large, interconnected polysaccharides that are composed of chains of two alternating sugar and cross bridges of amino acid. Scientists describe two basic types of bacterial cell walls. There's gram-positive and gram-negative. So, this image here shows the possible structure of the peptidoglycan. So again, peptidoglycan is composed of chains of NAG and NAM linked by tetrapeptide cross bridges, and in some cases, as shown here, connecting chains of amino acids to form a tough yet flexible structure. The amino acids of cross bridges differ among bacterial species. Okay. Gram-positive bacterial cell walls. Gram-positive bacterial cell walls relatively have thick layer of peptidoglycan. It contains unique chemicals called teichoic acid and lipoteichoic acid. Teichoic acid serves to stabilize the cell wall and hold it in place, whereas lipoteichoic acid anchors the thick layer of peptidoglycan to the cytoplasmic membrane. Gram-positive appear purple following the Gram staining procedure. This is because the thick cell layer of peptidoglycan traps the purple dye used in the Gram staining procedure. The Gram staining procedure is called the crystal violet dye. Up to 60% of mycolic acid in acid-fast bacteria help cells survive desiccation. Now, this this bullet is referring to how the presence of mycolic acid in the cell wall of these bacteria helps them withstand drying out. Desiccation basically means drying out. So this image here shows a gram-positive cell wall. As you can see, it has a thick layer of peptidoglycan, and the lipoteichoic acid anchors the cell wall to the cystoplasmic membrane. So the lipoteichoic acid, these are anchored into the lipid membrane and extend to the peptidoglycan layer. Okay. We then move on to gram-negative bacterial cell walls. Gram-negative bacteria only have a thin layer of peptidoglycan. It also has a bilayer membrane outside the peptidoglycan, which contains the phospholipid proteins and the lipopolysaccharides. So the bilayer refers to the two layers of leaflets. There's a inner leaflet and an outer leaflet. Now, in the inner leaflet, it composes of the phospholipids and proteins, whereas the outer leaflets consist of the lipopolysaccharides. So the lipopolysaccharide is an endotoxin, a toxic molecule composed of the lipid A. Now, the lipid A portion of the LPS can cause fever, vasodilation, inflammation, shock, and blood clotting. So when a G-negative bacteria dies, it releases lipid A. It may also impede the treatment of disease. So what this bullet point is saying is that the outer membrane of the G-negative bacteria can block certain antibiotics. And gram-negative bacteria appear pink following gram staining procedure. Okay. So this image here shows a gram-negative cell wall. So as you can see, it has a thin layer of a peptidoglycan and an inner leaflet composing of a phospholipid layer and proteins right here, and an outer leaflet composing of phospholipids, proteins, and lipopolysaccharide. Okay. Bacteria without cell walls. So a few bacteria lack cell walls, and they are often mistaken for viruses due to the small size and the lack of the cell wall. But they have other features of prokaryotic cells, such as ribosomes. So, bacterial cytoplasmic membrane. The structure is referred to as the phospholipid bilayer, which means it's a two-layered structure of a cell's membrane. It is composed of the lipids and associated proteins. Now, the integral proteins is embedded within the phospholipid bilayer, whereas the peripheral proteins are not embedded, and instead they are loosely attached. The fluid mosaic model describes the current understanding of the membrane structure. So the fluid mosaic means it is scattered in a pattern throughout the phospholipid bilayer. So here's an image of a prokaryotic cytoplasmic membrane, a phospholipid bilayer. So if you look at the structure here, a single phospholipid contains a hydrophilic head and a hydrophobic tail. And the way it's structured is that the interior of the phospholipid bilayer is hydrophobic, and that's because the tails are facing each other. But the outer layer of the hydrophilic head is hydrophilic because the head is hydrophilic, okay? Now, it also has a peripheral protein. Again, peripheral proteins are loosely attached, whereas the integral proteins are embedded within the phospholipid bilayer. Now, there's more factors as to how it's structured, but I'll go over those in a bit. So here's a video.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and 2-fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain two types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
Okay. So the function of the cytoplasmic membrane. Now, one of the functions is that it controls passage of substances into and out of the cell, which means it allows nutrients to enter the cell and waste to leave the cell, helping the cell maintain its necessary function. It also harvests light energy into photosynthetic bacteria, which means it harvests light energy to make ATP. The ATP is the cell's energy molecule. It is selectively permeable and naturally impermeable to most substances. This means that selectively permeable allows certain ions and molecules in, whereas it's impermeable to most substances, which means it doesn't pass large molecules, ions, and molecules with electrical charges in. So proteins allow substances to cross the membrane. So if an ion or a molecule is impermeable, they have to use proteins to cross the membrane. And the proteins function as pores, channels, or carriers that allow substances to cross the membrane. And it can be active, which means it uses energy, or passive, which means it doesn't use energy. It also maintains concentration and electrical gradient. Now, here are two quick analogies explaining concentration gradient and electrical gradient. So for concentration gradient, imagine you spray a perfume in one corner of a room. So initially, the perfume molecules are highly concentrated in that spot, but over time, they spread out to areas where there are fewer perfume molecules. The perfume will move from where it's more concentrated, which is the corner, to where it's less concentrated, which is the rest of the room, until it's evenly distributed. So that's concentration gradient. Now for electrical gradient, imagine you have a hill, and at the top of the hill, there are positively charged balls, while at the bottom, there are negatively charged balls. The hill represents the difference in charge. And just like how the gravity pulls object down a slope, the positively charged balls naturally want to roll down towards the negative ones to balance it out, because opposites attract. A positive and a negative attract each other. And then here's a video.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
Okay. Electrical potential of a cytoplasmic membrane. Now, the electrical potential is negative 70 millivolts because there is more negative charges inside the cell compared to the outside the cell. And here's another video explaining the principle of diffusion.
Passive transport includes diffusion, nonspecific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
Okay. Function. Passive processes. So what passive processes means is that the cell does not need to use ATP. And there's three types. There's diffusion, which means the net movement of a chemical down its own concentration gradient. Facilitated diffusion, which means the movement of substances across the cell membrane via a protein channel. And osmosis, in which is the diffusion of water molecules across a semi-permeable membrane. Okay. So the passive processes of movement across a cytoplasmic membrane. Again, diffusion of a small or lipid-soluble chemical through the membrane. So these are what you call a permeable substances, which means they can just go through without using any channel proteins. Facilitated diffusion of the several types of chemical through a non-specific channel. So this is non-specific, which means any molecule or ion can pass through. Facilitated diffusion through a permease of a specific chemical. This means that specific ion or molecule can only pass through the protein channel. Then you have osmosis. Osmosis is the net movement of water through a specific channel or through the membrane. As you can see, it can go through the membrane or through the phospholipid bilayer because they're semi-permeable. Osmosis, the diffusion of water across a semi-permeable membrane. So what is going on in the picture? Well, the left and right compartments contain solutions with solute, which is the red dot, and the solvent, which is just the water. Okay, the membrane between the compartments only allow water to move and not solute particles. So this membrane right here only allows water to move. So even though both compartments have the same osmolarity, which basically means concentration of solute, the right compartment has larger volume of solution because water can move to balance the concentration. So the water basically flowed from left to right to equalize the concentration, resulting in more water in the right compartment. This basically demonstrate how osmosis seeks to balance solute concentration by moving water, even if the solutes themselves can't pass through the membrane. Here. So the effect of isotonic, hypertonic, and hypotonic solution on a cell. So starting off with isotonic solution. Now, for cells without a wall, this one, the concentration of solutes inside and outside the cell is the same. So water movement is balanced and the cell maintains its shape. For cells with a wall, the same balance occurs. Water moves in and out equally, so the cell retains its usual shape. For hypertonic, in cells without a wall, water moves out of the cell due to the higher solute concentration outside, causing the cell to shrink. In cells with a wall, water also exits the cell, and the cytoplasm shrink away from the cell wall, but the cell wall remains intact. In hypotonic, in cells without a wall, water rushes into the cell because the solute concentration is higher inside, so the cell may swell and burst. In cells with a wall, water enters, causing the cell to swell, but the cell wall prevents bursting, resulting in a turgid cell. Okay. So here's another video explaining the special types of diffusion.
Simple diffusion does not require special membrane proteins for movement of molecules. However, other types of diffusion do require such proteins for the transport of molecules. Nonspecific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of nonspecific facilitated diffusion, these permeases allow a wide range of molecules that meet charge or size criteria to pass through their channel. Some facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of a specific facilitated diffusion, these permeases only allow specific types of molecules to pass through their channel. The channel contains a recognition element. If the molecule is not recognized, the channel will not open to allow the molecule to pass into the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through the phospholipid bilayer with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes is really the force that causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and can cause it to swell up and possibly burst.
Okay. So function, active processes. Now, active processes is the movement of a substance against its own chemical gradient via carrier protein and requiring cell energy from ATP. It is active transport, which means it uses energy. The group translocation is when a substance is chemically modified during the transport. So as a substance is transported into the cell, it is chemically modified in such a way that it becomes a different substance once it's inside. This process requires energy. Okay. So the mechanisms of active transport. We first start with the unit port. This mechanism involves the transport of a single substance in one direction across the membrane. If energy is required, ATP is used to move the substance against its own concentration gradient. Then antiport. This type of transport involves two substances being moved in opposite directions. one substance is transported into the cell while the other is transported out. And often, one of the substances move along its concentration gradient, which help power the movement of the second substance against its gradient. Then, the coupled transport. In this scenario, ATP is first used to pump a substance out the cell through a uniport. As this substance flows back into the cell down its own gradient, concentration gradient, it brings another molecule with it through a symport. This process is an example of secondary active transport, where the energy for the transport of the second molecule is derived from the electrochemical gradient set up by the primary active transport using ATP. Okay. Group translocation. So by definition, group translocation is the active process in which occurs in some prokaryotes by which a substance is being actively transported across a cell membrane and is chemically changed during the transport. So this slide shows the process in which it involves a chemical change to the substance being transported. In this case, the addition of phosphate. So here's the quick overview of what we just went over. Passive transport processes. This is the process that requires no use of energy by the cell. The electrochemical gradient provides the energy. Diffusion. Molecules move down the electrochemical gradient through the phospholipid bilayer of the membrane. And examples of transported substances are oxygen, carbon dioxide, and lipid-soluble chemicals. Facilitated diffusion. Molecules move down their electrochemical gradient through a channel or carrier proteins. And the examples of transported substances is glucose, fructose, urea, some vitamins. Osmosis. Osmosis is when water molecules move down their concentration gradient across a selectively permeable membrane. And what's being transported? Water. Active transport processes. This is when cell expends energy in the form of ATP to move a substance against its electrochemical gradient. Active transport. ATP-dependent carrier proteins bring substances into the cell. And the examples being transported is sodium, potassium, calcium, hydrogen, and chloride. Group translocation. The substance is chemically altered during the process. And the examples of the transported substances is glucose, mannose, and fructose. Cytoplasm of bacteria. Cytoplasm of the bacteria. Cytosol. Cytosol is the liquid portion of the cytoplasm. It is mostly water, but it also contains dissolved substances like ions, proteins, and sugars. It contains the cell's DNA in a region called the nucleoid. The nucleoid is the region of the prokaryotic cytosol containing the cell's chromosomes. Inclusions. Inclusion is the deposited substances such as lipid, gas vesicles, or magnetite stored in the cytosol of the cell, and it may include the reserve deposits of chemical. Such chemicals may be taken in and stored into the cytosol when nutrients are abundant and then utilized when nutrients are scarce. And then we have endospores. Endospores are highly durable dormant structure formed by the bacteria like the bacillus and the clostridium in response to harsh conditions. It is not for reproduction, but for survival. Endospores have unique structures produced by some bacteria. It is a defensive strategy against its unfavorable conditions. The vegetative cells transform into endospores when multiple nutrients are limited. So, the process of endospore formation is called the sporulation. It is resistant to extreme conditions such as heat, radiation, and chemicals. So here's the formation of an endospore. So step 1 is DNA replication. The vegetative cell replicates its DNA in the preparation for creating an endospore. Step 2, formation of the forespore. The cytoplasmic membrane of the vegetative cell begins to invaginate or fold inward to form the forespore, which will eventually become the endospore. This separates the replicated DNA into the forespore area. Step 3, engulfment of the forespore. The cytoplasmic membrane continues to grow and engulfs the forespore, forming a second membrane around it. This creates a protective barrier. At this stage, the DNA of the vegetative cell outside the forespore disintegrates because it is no longer needed. Step 4, cortex formation. A thick cortex made of peptidoglycan is deposited between the two membranes surrounding the forespore. Dipicolonic acid and calcium ions accumulate inside the developing endospore, which helps make it resistant to heat and chemicals. Step 5, spore coat formation. A tough spore coat forms around the endospore, adding another protective layer that contributes to its resilience. Step 6, maturation of the endospore. Thus, the spore coat matures, and the endospore increases in resistance to extreme environmental conditions like heat, chemical, and radiation. And step 7, endospore release. The vegetative cell around the endospore eventually dies, and the endospore is released to the environment. It can remain dormant and resistant to harsh conditions until it is ready to germinate into a new vegetative cell when conditions improve. So the formation of endospores usually occurs over a period of 8 to 10 hours. So the image here shows the transmission electron microscope of an endospore in a bacillus or a clostridium. And in this case, the image is showing an endospore inside a vegetative cell of a bacillus. Cytoplasms of eukaryotes, non-membranous organelles. Now, non-membranous organelles are structures that are not surrounded by membranes, but they still perform important functions within the cell. One of them includes ribosomes. Ribosomes is the site of protein synthesis in which it assembles amino acids into proteins. It is composed of polypeptides and ribosomal RNA. Prokaryotic and eukaryotic ribosomes have structural differences. Prokaryotes have 70S ribosomes, whereas eukaryotes have 80S ribosomes. So prokaryotes are smaller, whereas eukaryotes are bigger. So the size of ribosomes are expressed in Svedbergs, which is the S right here. Now, why is this important? Well, many antibiotics can stop bacteria from making proteins by blocking their 70S ribosomes. And since humans have 80S ribosomes, the antibiotic doesn't affect human cells. So the second one is cytoskeleton. The cytoskeleton is an internal scaffolding made of protein fibers that gives the cell its structure plus carry out important functions. It is composed of 3 or 4 types of protein fibers and can play different roles in the cell. For example, it can play cell division, cell shape, segregate DNA molecules, and move through the environment. So this image here shows a rod-shaped bacterium called the Bacillus subtilis, in which it has a helical cytoskeleton composed of only a single protein, which has been stained with a fluorescent dye. Okay. So tell me why. The 2001 bioterrorist anthrax attacks in the US involved the Bacillus anthracis. Why is B. anthracis able to survive in the male? And the answer to that question is that the B. anthracis survives in the male because of its endospores. The endospores are resistant to harsh conditions, so they can remain dormant for long periods. And when they find the right condition, like finding a human body, they become active again, allowing them to cause infections. This durability makes B. anthracis an effective agent in the 2001 bioterrorist attack. Archaeal cell walls and cytoplasmic membranes. So, most archaea have cell walls, but they do not have peptidoglycan. It contains a variety of specialized polysaccharides and proteins. All archaea have cytoplasmic membranes, in which it maintains electrical and chemical gradients. It also controls the import and export of substances from the cell, using the membrane protein as ports and pumps, just as proteins are used in bacterial cytoplasmic membranes. Okay, so this slide shows the representative shapes of archaea. So image A right here shows a cocci, a Pyrococcus furiosus, and it is attached to a rod shape called the Methanopyrus canthari. This one. Image B shows an irregularly shaped archaeon. It is called the Thermoplasma acidophyllum. For image C, this shows a square archaeon, and it is called the Halo quadratum Walsby. Cytoplasms of the archaea. Archaeal cytoplasms is similar to bacterial cytoplasms in which it has a 70S ribosome, it has a fibrous cytoskeleton, and it has a circular DNA. So, like bacteria, the cytoplasm of the archaea is like a gel-like substance carrying essential cellular components. And with circular DNA, it is suspended in a liquid part of the cytoplasm called the cytosol. However, archaeal cytoplasms also differ from bacterial cytoplasms, in which it is different ribosomal proteins, different metabolic enzymes to make RNA, and different genetic code more similar to eukaryotes. External structure of eukaryotic cells. Glycolysis. So glycolysis are absent in eukaryotes that have cell walls, like plants and fungi. It is not as organized as prokaryotic capsules. It helps anchor animal cells to each other. It also strengthens the cell surface. It provides protection against dehydration, and it functions in cell-to-cell recognition and communication. Eukaryotic cell walls and cytoplasmic membranes. So, fungi, algae, plants, and some protozoa have cell walls. They rely on these structures for protection, support, and maintaining their shape. It is composed of various polysaccharides. So, for example, cellulose is found in plant cell walls. So, what this is saying is that cellulose is the primary structure component of plant cell walls. Fungal cells is composed of cellulose, chitin, and or glucamon. glucomannan. And algal cell walls is composed of a variety of polysaccharides. So substances like cellulose, proteins, agar, carrageenan, silicates, algin, calcium, carbonate, or a combination of these. Endocytosis is an active transport process. It is used by some eukaryotic cells in which pseudopods surround a substance and move into the cell. So this image here shows a pseudopod. So the pseudopod extends to surround the solid and or liquid nutrients, which become incorporated into a food vesicle inside the cytoplasm. Active transport processes found only in eukaryotes, endocytosis and exocytosis. So endocytosis, there's two processes. There's phagocytosis and pinocytosis. So a description of these two, or yeah, actually these two, is that substances are surrounded by pseudopods and brought into the cell. Phagocytosis involves a solid substance, and pinocytosis involves liquids. So the examples of transported substances is bacteria, viruses, age, and dead cells for phagocytosis, whereas pinocytosis is liquid nutrients in extracellular solutions. Then we have exocytosis, which is basically the reverse process of endocytosis. A description is that the vesicles containing substances are fused with cytoplasmic membrane, dumping their contents to the outside. And the examples of transported substances is waste and secretions. So what we have to know here is that phagocytosis engulfs a solid, and pinocytosis engulfs a liquid. So tell me why. Many antimicrobial drugs target bacterial cell walls. So why aren't there many drugs that act against bacterial cytoplasmic membranes? And the answer to that is, since humans don't have cell walls, the antimicrobial drugs will not target our cells. However, if drugs targeted bacterial plasma membrane, then they will also kill our plasma membranes as well. All right. So cytoplasms of eukaryotes. We have flagella, structure and arrangement. So flagella in eukaryotes differ structurally and functionally from prokaryotic flagella. So eukaryotic flagella is located within the cytoplasmic membrane. So this means that the flagella is located inside the cell, meaning the cytoplasmic membrane surrounds them. The shaft is composed of tubulin arranged from microtubules. So think of the microtubule in the eukaryotic flagella like a bundle of straws. There's 9 pairs in the circle outside and 2 single straws in the middle. More information into that in a bit. Filaments anchored to the cell by basal body, and there's no hook, unlike prokaryotic flagella. And it may be single or multiple, and it is generally found at one pole. So when they say that it may be single or multiple, this refers to the flagella appearing as either a single structure, which is just one flagellum, or as multiple structures, several flagella in a eukaryotic cell. And when they say that it is generally found at one pole, it refers to the fact that flagella are usually located at the particular end or the pole of the cell. So this image here shows a eukaryotic flagella and cilia. So this is a micrograph of a protozoa known as the Euglena, which possesses a long flagellum right here. So continuing on with flagella, and we go into the functions. So eukaryotic flagella move differently from those of prokaryotes. So what this means is that they don't rotate like bacteria from prokaryotic cells. It moves in a smooth wave-like motion called pendulation. It's bending slash flexing in a rhythmic wave-like pattern. So an example of this is like a sperm cell. They either push or pull a cell through the medium. Push meaning it moving in a way that propels the cell through its environment, and pull in which the flagella pulls the cell through the medium by moving in the opposite direction, similar to how a rope may pull a boat through the water. Okay, so here shows an image of a flagella. Now, the eukaryotic flagella undulate in waves that begin at one end and traverse at the length of the flagellum. Okay, now we move on to cilia. Cilia are shorter and more numerous than flagella. The coordinated beating propels cells through the environment, meaning that it moves in a synchronized rhythmic pattern, working together to push slash pull the cell through its environment. It is also used to move substances past the surface of the cell. So, for example, in the human respiratory system, cilia moves in wave-like motion to help clear out the dust plus microorganisms from the airway, keeping the lungs clean. And here shows an image of a cilia. So the scanning electron micrograph of a protozoa named Blephorrhema, which has numerous of cilia. Okay. So here shows the movement of a cilia. Now, cilia move in a power stroke, which is then followed by a return stroke. So in the power stroke, a cilium is stiff. Then it relaxes during the return stroke. Okay. So other non-membranous organelles. We have ribosomes. Again, for prokaryotic ribosomes and eukaryotic ribosomes, they basically have the same function, in which they are the instructors that produces proteins by linking amino acids together. However, eukaryotic ribosomes are larger than prokaryotic ribosomes, in which eukaryotic ribosomes are 80S compared to prokaryotic ribosomes, which is 70S. It is composed of the 60S and 40S subunits. So reading that statement might be confusing, because how come 60S plus 40S does not equal to 100S? So let me give you an analogy explaining as to why the ribosome subunit add up to 80S instead of 100S. So imagine you have two objects, a large book and a small box. If you were to drop each each object into the water individually, they would sink at different rates depending on their shape, size, and how they interact with the water. So then let's say the large book sinks at one speed, let's say 60 units at this case, and the small box sinks at another speed, say 40 units. Now, if you take the book and the box together and you drop them into the water, you wouldn't expect their sinking speed to be simply the sum of the individual speeds. This is alluding to the fact that 60 plus 40 equals 100. So instead, their combined shape and interaction with the water changes, and they would sink at different rate, maybe at 80 s units. So hopefully that made sense. We then have the centrioles and centrosome. So for centrioles, they are cylindrical structures made up of microtubules that are important for organizing cell division. Then there's also centrosome. Centrosome contain the centrioles and act as organizing center for microtubules in the cell. Even though plants and most fungi lack centrioles, they still have the centrosome-like region to manage their microtubules. So centrioles play a role in mitosis, cytokinesis, and the formation of flagella and cilia. Centrosome is a region of the cytoplasm where centrioles are found, and they are not found in all eukaryotic cells. So, for example, animal cells and some fungal cells, um, they have centrioles and centrosomes, whereas plants, algae, and most fungi do not have centrioles and centrosomes overall. Okay, so a centrosome is a region of cytoplasm that in animal cells contain two centrioles at right angles to one another. Each centriole has 9 triplets of microtubules. Image A shows the transmission electron micrograph of a centrosome and centrioles. Then image B shows the rendition of a centrosome. Okay. Now we move on to membranous organelles. A membranous organelle refers to an organelle within a eukaryotic cell that is surrounded by a membrane. We first have the nucleus. So the nucleus is like the control center of the cell. It is often the largest organelle in the cell and is usually in a spherical or oval in shape. It contains most of the cell's DNA. The semi-liquid portion called the nucleoplasm is inside the nucleus. The nucleoplasm is a semi-liquid matrix inside the nucleus. It is similar to how cytoplasm is the semi-liquid inside the whole cell. And inside the nucleoplasm, it contains the chromatin. A chromatin is a thread-like mass of DNA. It is associated with histones, which help to package nuclear DNA. RNA is synthesized in the nucleoli present in the nucleoplasm. Nucleoli are specialized regions where RNA is synthesized and ribosomes are assembled. It is surrounded by a nuclear envelope. A nuclear envelope is a double membrane surrounding the nucleus. Now, in the nuclear envelope contains the nuclear pores. The nuclear pores are protein-lined channels that pierce the double membrane. It contains the import/export of substances through the envelope. So here shows a eukaryotic nucleus. Now, this is a micrograph in an artist's conception of a nucleus, in which it shows the chromatin, the nucleolus, and the nuclear envelope. The nuclear pores punctuate the two membranes of the nuclear envelope. Okay. And then we move into the endoplasmic reticulum, or the ER. The ER traverses the cytoplasms of eukaryotic cells and functions as transport system. Its net-like arrangement of flattened, hollow tubules is continuous with nuclear envelope. And it has two forms. one being the smooth endoplasmic reticulum, or the smooth ER. It is referred to as the smooth endoplasmic reticulum is because of its smooth surface. The smooth surface lacks ribosome on its membrane. The smooth ER plays a role in lipid synthesis as well as transport. We then also have the rough endoplasmic reticulum, or the rough ER. It is rough on the surface because it is stubbed with ribosomes on its surface. The rough endoplasmic reticulum produces protein, which are then inserted to the lumen, or the inner space of the rough endoplasmic reticulum. From there, the proteins can be modified, folded, and transported to other parts of the cell, or packaged for secretion outside the cell. So here's an image of the endoplasmic reticulum. It functions in transport throughout the cell. The ER functions, yep. So here's a picture of the endoplasmic reticulum. Now note the differences between the smooth ER and the rough ER. The smooth ER is smooth because it lacks ribosomes. The rough ER is rough because it has ribosomes. And the ER functions in transport throughout the cells. And so then we move into the Golgi body. The Golgi body is like the shipping department of a cell. It receives, processes, and packages large molecules for export from the cell. Packaged molecules in secretory vesicles that fuse with cytoplasmic membrane. So in other words, it transports processed molecules to the cytoplasmic membrane. It is composed of flattened hollow sacs surrounded by phospholipid bilayer. Not all eukaryotes have Golgi body. Some cells may not require exclusive process and packaging of molecules, so they do not have to have a Golgi body. So here's an image of the Golgi body. A Golgi body is composed of a flattened sacs. The proteins synthesized by the ribosomes on the rough ER are transported via vesicles to a Golgi body. The Golgi body then modifies the proteins and sends them via secretory vesicles to the cytoplasmic membrane, where they can be secreted from the cell by exocytosis. And so then, we move into lysosomes, peroxisomes, vacuoles, and vesicles. So these four are membranous sacs that functions to store and transfer chemicals within the eukaryotic cell. So starting off with vacuoles. Vacuoles are large sacs that store substances like water, starch, lipids, etc. It is so large, in fact, that the rest of the cytoplasm is pressed against the cell wall in a thin layer. We then go to vesicles. Vesicles are smaller sacs that help transport materials within the cell. It goes from the ER to the Golgi to the cell membrane. Now, these four store and transfer chemicals within the cell. It may store nutrients in a cell. So we first have the lysosomes. Lysosomes are vesicles in animal cells that contain digestive enzymes. So lysosomes contain catabolic enzymes, meaning that it helps break down large complex molecules into simpler ones. We then have peroxisomes. Peroxisomes are vesicles found in all eukaryotic cells that degrades poisonous metabolic waste. So again, it contains enzymes that degrade poisonous waste. Degrade meaning break down. So here's an image of a vacuole. The large central vacuole of a plant cell, which constitute a storehouse for the cell, presses the cytoplasm against the cell wall. Okay. So this image here shows or illustrates the processes of endocytosis, specifically phagocytosis, and exocytosis in a eukaryotic cell. It shows how a cell ingests large particles, such as bacteria, and breaks them down, as well as how waste or processed materials are secreted from the cell. So going step by step. We first have the endocytosis or the phagocytosis. Phagocytosis is a type of endocytosis where the cell engulfs large particles like a bacteria. The cell membrane surrounds the bacterium, forming a vesicle known as the phagosome, or the food vesicle, which contains the ingested material. The phagosome fuses with a lysosome, forming a structure called the phagolysosome. The lysosome contains digestive enzymes that break down the ingested bacterium into smaller components that can be reused by the cell. The Golgi body processes molecules such as enzymes and proteins, which are packaged into secretory vesicles. These vesicles can form lysosomes that are used to digest foreign materials like bacteria. Once digestion is complete, waste products and undigested materials are packaged into secretory vesicles. These vesicles move to the cytoplasmic membrane, where they fuse with the membrane and release their contents outside the cell. This process is known as the exocytosis, which is a term used to describe secretion and elimination of waste. And then we move on to the mitochondria. The mitochondria is spherical or elongated in shape. It has two membranes composed of a phospholipid bilayer. So there's 2 layers right here, another 2 right here, making it 4 layers in totals. Um, the inner bilayer forms numerous folds called the cristae that increases the inner membrane surface area. And as you guys know, the mitochondria is known as the powerhouse of the cell. So therefore, they produce most of the cell's ATP. The interior matrix contains 70S ribosomes and a circular molecule of DNA. So the interior matrix is the innermost compartment of the mitochondria. It is surrounded by the inner membrane. It is the space where various chemical reactions occur, including energy production. The 70S ribosome refers to the size and the type of ribosome. And when it says circular molecule of DNA, it's referring to that the mitochondrial DNA is organized in a closed loop or a circular form rather than being a linear like the DNA in the nucleus of a eukaryotic cell. So here's the image of the mitochondrion or the mitochondria. As you can see, the inner membrane is folded into cristae that increases its surface area. So the chemicals involved in aerobic ATP production are located in the inner membranes of the mitochondria. The increased surface area provides more space for more chemicals. And then, and then we move on to the chloroplast. The chloroplast is the light harvesting structure found in photosynthetic eukaryotes. It capsules light energy and uses it to produce ATP and sugars via photosynthesis. Uses light energy to produce ATP. It has 2 phospholipid bilayer and a DNA. Just like the mitochondria and the nucleus, it has 4 layers because it has 2 phospholipid bilayer. There's a layer right here, another layer right here, and there's 4 layers in total. And the DNA. The chloroplast DNA is linear, similar to the DNA in the nucleus. And it has 70S ribosomes. Again, this refers to the size and the type of ribosome. So here's the image of a chloroplast. The chloroplasts have an ornate internal structure that are designed to harvest light energy for photosynthesis. And here shows the table of the functions of non-membranous and membranous organelles and their distribution among prokaryotic and eukaryotic cells that are summarized in this whole table. Yeah. And then we move on to the endosymbiotic theory. The endosymbiotic theory proposes that eukaryotes were formed from phagocytosis of small prokaryotes by larger prokaryotes, forming organelles. So the theory suggests that mitochondria and the chloroplasts in eukaryotic cells have evolved from ancient bacteria. A long time ago, a large cell engulfed a smaller energy-producing bacteria, and instead of being digested, these smaller bacteria started living inside the larger cell. Over time, the smaller bacteria became dependent on the larger cell and turned into the mitochondria, which makes energy, or the chloroplasts, which help with photosynthesis. This explains why the mitochondria and chloroplasts have their own DNA, ribosomes, and double membranes like bacteria. So the eukaryotes form from union of small aerobic prokaryotes with larger anaerobic prokaryotes. The small aerobic prokaryotes referring to the early bacteria that use oxygen for energy production, and the larger anaerobic prokaryotes referring to the ancient bacteria that relied on anaerobic respiration or fermentation for energy production. Um, the smaller prokaryote became the internal parasite. The parasite lost the ability to exist independently. Larger cell became dependent on parasites for aerobic ATP production. Aerobic prokaryotes evolved into mitochondria, and it is a similar scenario for the origin of chloroplasts. So basically the same thing that I said right here. And then finally, we move on to the comparison of archaeal, bacterial, and eukaryotic cells. So we start off with nucleus. Nucleus is absent in archaea, bacteria, but it is present in prokaryotes. We then have the free organelle bound with phospholipid membranes. It is absent in archaea, it is present in a few bacteria, and it is present in all eukaryotes. It is various types of present in all, including the ER, Golgi bodies, lysosomes, mitochondria, and the chloroplast. So we then go to glycocalyx. It is present in all archaea. For bacteria, it is present as organized capsule or unorganized line layer. And for eukaryotes, it is present in some, e.g. surrounding some animal cells. For motility, it is present in some archaea, some bacteria, and present in some eukaryotes. Some have flagella, cilia, or pseudopods. For flagella, some have flagella for archaea, in which it is composed of the basal body, hook, and filament, and the flagella rotates. And for prokaryotes, some have flagella, in which each composed of the basal body, hook, and filament, and the flagella rotates too. And for eukaryotes, some have flagella or cilia composed of a 9+2 arrangement of microtubules, and the flagella and cilia undulate. Then we move on to the cilia. It is absent in all in archaeal and prokaryotes, but it is present in some eukaryotes. We then move on to the fimbriae or pili. It is present in some archaea, present in some prokaryotes, and it is absent in all eukaryotes. Then finally, we have hami. It is present in some archaea, it is absent in bacteria, and it is absent in eukaryotes. Then we have the cell wall. The cell wall is present in most archaea, but it lacks peptidoglycan. For bacteria, it is present in most, but it is composed of peptidoglycan. And for eukaryotes, it is present in plant, algae, and fungi at cell wall. Cytoplasmic membrane. It is present in all. Archaea, bacteria, and eukaryotes, they all have a cytoplasmic membrane. Cytosol 2, it is present in all. Inclusions. It is present in most archaea, it is present in most bacteria, and it is present in some eukaryotes. And then we move on to ribosomes. Archaea and bacteria, they have the same size. They're 70S, Sbergs. But for eukaryotes, it is large in cytosol and the ER, but it is smaller on the mitochondria and chloroplasts. Finally, chromosomes. For archaea, it is common, single, and circular. For bacteria, it's also the same. It is common, single, and circular. And for eukaryote, it's linear and more than one chromosome per cell. And that should be it with the video, guys.